Okay, everybody, we're going to get started. So we have with us today Dr. Eva Feldman, who comes to us from Ann Arbor, where she is Russell DeJong Professor of Neurology. She received both her Ph.D. in neuroscience and her medical degrees from the University of Michigan, did her residency in neurology at Johns Hopkins Hospital, and returned to the University of Michigan for neuromuscular fellowship, remaining there ever since. And she is currently the director of the Neuropathy Center, director of research of the ALS Center, and director of the Alfred Taubman Medical Research Institute. Her basic and clinical research focus has primarily been investigating the pathogenesis and treatment of diabetic neuropathy and stem cell research for the treatment of ALS. She has multiple NIH awards and been on many committees, and most recently was elected to the National Academy of Medicine. And notably, her committee memberships they're noted for, she is a past president of the American Neurological Association. And she's going to talk to us today about intraspinal stem cell transplantation in ALS, clinical trials update.
Okay, so let's see. I was told I needed to do this and look in that screen. Is that okay? Okay, great. Well, thank you so much for having me. I've really enjoyed my visit. I really just had a lot of fun right now with the residents. As I mentioned to the residents earlier, my daughter is a brand new faculty member here. She joined in July. She's in the Department of Medicine as an assistant professor. So I was really motivated to come not only to see your department, and I was hoping to meet with your chair, who's a good friend, and also, to see the medical school and your hospital system to understand, where my daughter would be working. And this morning, as I just already mentioned the story, this morning as I went to Swedish to give grand rounds, I did not realize that Swedish was not the private hospital for UW, which is what I asked them, which is what I assumed they were. So it's really a very interesting, complex medical milieu here, I will have to say. And as an outsider, I don't think I understand it yet after a day. I'm very thrilled, though, to be here. I'm a very big fan of your chair. I think he's done a great deal for academic medicine. He's a great man. So what am I going to talk about today? Well, stem cell therapy and motor neuron disease, something I'm very passionate about. And as I was telling the residents, currently this is an era of team science. One person can really not do it all anymore. And so as you'll see, this has really been an effort. My colleagues at the University of Michigan, Dr. Gottman, who's a neurologist, Dr. Patel, who's a neurosurgeon, my colleagues at Emory University, John Glass, who many of you might know, a neurologist, and Nicholas Boulis, a surgeon. My colleagues at Harvard, Merit Kudowitz, who's the chair now of neurology and an ALS physician, and her younger colleague, Dr. Atassi. We've been funded primarily by the National Institutes of Health and the ALS Association. We also received funding from a small biotech, Neurostem, that helped develop these stem cells and are currently now funding a good portion of the clinical work. I take no monies from Neurostem, so I have nothing to disclose. So this is the journey I'm going to tell you about. It's kind of exciting, I think. And this is, how do you take an idea from your laboratory and take it from the laboratory for a disease you're passionate about and get it into clinical trials? And so what this is, let's see, here we go. So what this is the kind of typical time frame one does that, getting an idea and taking it through until one achieves an FDA trial. So what I'm going to do today is talk a little bit about the small animal validation that we did for ALS with our idea of using stem cells as a therapy. I'll tell you about that. Let you see some of the large animal validation. I'll introduce you to some of my favorite mini pigs. And then I want to spend most of my time talking about the human trial. And because I think that, in a way, as clinical and practicing neurologists in this room, is what most of us are very passionate about. And so the disease that we're going to focus on today is amyotrophic lateral sclerosis, or Lou Gehrig's disease. And as you can see here in this video, here is Lou Gehrig, really probably one of the premier baseball players ever to live, known as the Iron Horse. He himself became very frustrated with how he was performing Look, you can see his frustration there as he throws the bat down. And what you're going to see next is his very, very last time at bat, right there. He struck out. He took himself out of the game because he knew he wasn't doing well. Everyone else thought he should continue playing. He got on a plane. He went to Mayo Clinic, where he was diagnosed by a University of Michigan trained neurologist, I will tell you that, with the disorder that now carries his name, amyotrophic lateral sclerosis, frequently called Lou Gehrig's disease. And quite simply what this disorder is a degeneration of the large motor neurons in both the brain and the spinal cord. Now it's really much more complex than that, but really today's talk really isn't on the pathogenesis of ALS, which many of us are very passionate about, but rather on kind of translating a therapy to treat a disorder. So here is just a schematic of the spinal cord, and here you see in the ventral horn a blow up of the very large motor neurons, these beautiful motor neurons that of course emit axons that connect to muscles that allow us to move. And here what we see here is a stain for death-inducing proteins that turn the motor neurons brown, and these are motor neurons from an animal model of ALS that are undergoing an apoptotic or a death due to the disease. Now, there are many ways to tackle this disease, but before we talk about it, let's go back to Lou Gehrig. And look at that, very interesting. So here's an analysis of Lou Gehrig's batting averages, okay? And here's the analysis then in his final year at bat, when he was final year as a Yankee. And look, of course, what happened to his batting averages. Here's a picture of his hands, of his forearms from the Mayo Clinic archives. Isn't that remarkable? And then, of course, you see his picture up there. So, I mean, clearly he wasn't doing well batting because he had all that significant wasting and weakness. Now, a little anecdote, a very interesting story about Lou Gehrig is that he entered a clinical trial when he was at the Mayo Clinic. It was a kind of landmark trial. It was using vitamin C for the treatment of ALS. And he was certain within the first 6 weeks that he had been cured of the disorder, and as were his physicians. So the placebo effect and our desire to get better with clinical trials is certainly not a new phenomenon. It certainly existed in the 30s. He actually died about 6 months after being diagnosed at Mayo Clinic and entering that clinical trial. So, as I alluded to, there are really two ways to approach working with this disorder and creating new therapies. The best way, by far, bar none, is a mechanism-based approach. And I'm going to tell you a little story about why I veered away from that approach to do stem cell therapy. But mainly, it's because that approach is somewhat limited by our lack of understanding of the clear pathogenesis of the disorder. We know that about 18 to 20% of patients have an inherited disorder that probably has to do with RNA metabolism, the C9 open reading frame 72 gene mutation, but the other 80% or so are really still sporadic ALS. And when we say ALS, we just wrote a small editorial, and it was entitled ALS, not one disease but many, because it carries one name, but it really is multiple different phenotypes. So the other way to tackle it is to say, look, I don't quite understand why these motor neurons are dying, but let's just use what I'll call a cellular approach. Let's put stem cells in this area and see if they'll provide neuroprotection. Maybe they'll form synapses on the sick motor neurons, maybe they'll make growth factors, maybe they'll change the inflammation, maybe they'll do nothing and not work at all. So we took the current approach, the stem cell approach, for this trial in ALS. But I think, again, for the junior faculty, a good question is, why did I make that change for this particular disorder? So I spent the first 10 years of my career working on a growth factor known as IGF-1, also known as myotrophin, and we took it from the bench, as I showed you in that very first slide, all the way to a large phase 3 clinical trial, 330 patients, placebo-blind, and we'd actually done two phase 2 trials that had looked fairly promising, and the company had done one phase 3 trial that looked promising, but this was an NIH-funded trial, 2 years duration, unheard of it really in ALS trial world. And I was sure it was going to work, right? I mean, I just knew it. I knew it from all my preclinical data in my heart. I knew it was going to work. And I remember, like it was 2 minutes ago, opening up a big envelope. It was literally an envelope I got from the FDA with the trial results. And here's the placebo control line for our primary endpoint, and here's the IGF-1 injected control line for the primary. They were so superimposable. At first I had to see that there was one line or two. So that was a clear failure in terms of bringing a therapeutic, but it also brought an opportunity. So for the young assistant professors in the audience, failures just give you a chance to kind of retool and think, okay, that wasn't the right way to go, so how can I do it? So that's why we decided to try stem cells. And so that's how I got into the stem cell business. So, and the idea that I took was very simple. This was extremely simple. And that is, here we have a diagram of a control spinal cord. The idea being in an ALS spinal cord, I've already told you, is what I'll call a diseased environment. And all we're going to do is put stem cells in the ventral horn area of the spinal cord and see what happens in a animal model of ALS. But step one, what type of stem cells, right? So which one would provide the most therapeutic benefit? Another story about of how I went about it, and another error, as you'll see. So I was certain that it was going to be embryonic stem cells. I thought embryonic stem cells are totipotent. They'll get into that environment. They'll be able to, offer the most protection. They'll be able to, form neurons, form glial cells, abrogate the inflammation. So we actually set off and became very invested in forming embryonic stem cell lines. And I thought I'd go over with you how we formed embryonic stem cell lines. Probably you're very familiar with it, but just in case if there are any students in the audience. So as you know, in vitro fertilization clinics, man donates sperm, woman donates an egg, and in a petri dish we form embryos. And at Michigan we have a very large fertilization clinic, and those embryos that aren't going to be used, if the parents consent, we can make embryonic stem cell lines out of those embryos. And how do we do it? Well, what happens is after 5 days here, this is known as the human blastocyst, and this is known as the inner cell mass. And what we do is we take this inner cell mass, we really disperse it into a petri dish, and then those cells are truly totipotent. And within a few months, you can have from these 100 cells, you can literally have tens of millions of cells. Now, as you do know, and this becomes important, if you allow the embryo to continue to develop, you get the formation, as you see here, of the 3 germ layers, and then more progenitor cells that will only become one type of cell or another. So they're not totipotent; they're called pluripotent. And here's an actual human blastocyst, and here's an example of what the inner cell mass looks like. So now let's just look at us creating an embryonic stem cell line. So here we have a human blastocyst, and the first thing we need to do is we pull the outside covering off of the human blastocyst so we can easily then get to the inner cell mass. Then we take a laser and we make us just a slight laser cut here so that we end up here with our inner cell mass. And so then we'll take this inner cell mass and we'll disperse it. It's only a few hundred cells, and we'll disperse it. And then what we get really quite quickly are these large embryoid bodies. These are hundreds of thousands of stem cells in these large embryoid bodies. And then we could, redisperse them, regrow them, so we can culture. And we have in the bank many stem cell lines. Interestingly, and as an aside, we also have many disease-specific stem cell lines, which I think are very interesting and available to anyone if they're interested, including the C9ORF72 line. So I've got my stem cells, right? I've let IGF-1 go down, go its merry way, and I started injecting stem cells into the spinal cord of rats. And what happens? Normal rats, just to see if I can do it. So here's the spinal cord of a normal rat that had been injected with stem cells, but here's a spinal cord of another rat that had been injected with embryonic stem cells. And here's a higher power. And as I'm not sure you can see here, I'm sure you can see here well, there's really significant tumor formation. as you see here, and this was actually the formation of a bone. So we had 90%, 95% of the time, the animals did great. 5% of the time we had bone, we had a tooth, we even had hair once, I couldn't believe it. So these are really totipotent cells. They can make absolutely anything. So back to square one. So stem cells in and of themselves, and I think it's important to understand this because you'll frequently ask this in clinic too. Embryonic stem cells really at this point aren't ready for prime time for transplantation. They need to be helped or differentiated in some way. So we turn to using neuroprogenitor cells. So remember how I told you if you let the embryo go longer, you can have cells or progenitor cells from specific cell lines. So that's what we did. We actually elected at that point to take a step back and look at the entire landscape of stem cells and see what was out there that was human, that was NIH approved, that had some background to it that we could begin to try in our animal models. And we discovered a stem cell line that had been made at the National Institutes of Health about 10 years ago from an 8-week-old fetus. And what they had done, the fetus had been donated to the NIH, and they had taken cells from the spinal cord area of the fetus and created a stem cell line that they called the human spinal stem cell line. And they had then licensed that stem cell line out to a small biotech known as Neurostem. And so we approached Neurostem, and they were actually very excited to work with us. Could we try their cells in an animal model of ALS? And what can you tell me about your cells? Well, they could assure us they didn't make tumors because they were about to do a clinical trial in spinal cord injury. So they had done all the rat preclinical tumor genesis work. And they could tell us that they turned into neurons, astrocytes, and oligodendroglia. So we were now on our way with the new stem cell line. And the first experiment was very, very simple. And that is what we had done in the previous rats, actually, is we did a laminectomy, so we exposed the spinal cord. And then all we're doing is injecting stem cells. But in this case, we're injecting into the diseased spinal cord of an ALS animal of an ALS rat. And so there are multiple different animal models of ALS, but the main one is a rat that carries a mutation for something known as superoxide dismutase 1 or SOD1 rat. So that is the animal model that we used. And here's some great data. Finally, a home run, okay? So here I'm showing you a low power figure where you can see the stem cells, which in this particular experiment are going to be green. And here's a higher power figure of stem cells that have been injected, again, in the spinal cord of an ALS rat. And what I want you to see are a couple of things. One is a proportion of them become beautiful neurons and put out axons, as you can see here, okay? And they integrate well into the cord. So this was, I think, really our first, home run. And I will also tell you that we did about 50 animals with no tumors, although we were told they couldn't make tumors. So we finally had stem cells that would get into the spinal cord, would integrate, would turn into neurons without tumors. So we're very excited, and I did this in collaboration with Dr. Martin Marsala at University of California, San Diego. I decided after my IGF-1 experience, I needed a few months off, so I took a sabbatical in January and February, March, 3 years in a row to San Diego from Michigan. One might question my choice of months. And so let me show you some other stuff, some other data, cool data that we got. Working now with Dr. Koliestos from Johns Hopkins, Dr. Joe from Neurostem, who supplied for us the cells. And so here is an example. Again, now this is this SOD1 rat, this ALS rat. And I just want you to look here for starters. We either injected into those rats live stem cells, or what I did is we cooked the stem cells. Probably not the best in retrospect, but again, these were early days. We literally cooked the stem cells and injected them, so we called them control grafts. They had no, viability. And look what happens. We see a preservation of the motor neurons here. These are the endogenous motor neurons of the ALS rat. So instead of dying, the way I showed you they did do, okay, early on in the talk, they lived, okay, and compared to those rats that received the baked stem cells. And if you look, we can actually quantitate the number, and you see a marked improvement in the motor neuron number. And in parallel, what we saw was a nice increase in rat survival. And actually, we had to put these rats down at this point because they could not easily right themselves, but they were based on this UCSD protocol. But in other experiments that were done at another institution, they didn't have such stringent animal protocols, which maybe was good, maybe was bad. The rats lived another 10 to 15 days. So we had survival. I mean, that's the key. We had survival, and we actually had a mechanism. I mean, that there were more motor neurons there. So I'm now going to tell you, I'm going to show you one slide and talk about 5 basic science papers, because that's not why we're here, is just look at a bunch of Western blots and PCR and immunohistochemistry. So let me tell you what these stem cells do. It's really interesting. So we inject them into the diseased cord, right? What they do is they surround those sick motor neurons and they make synaptic contacts. It's really interesting. And they particularly like to go to the sick neurons. They make contacts. Secondly, they produce a bucket of growth factors. And irony of all irony, the growth factor they produce the most is, right, it's IGF-1, of course, which I thought was just like amazing. And then besides that, there's a lot of inflammation in neurodegenerative diseases, ALS, Alzheimer's, all of them. All the inflammation is abrogated. It is all gone. They are really highly anti-inflammatory. And these are all characteristics that other groups have now seen also with these stem cells. But we were, of course, the first to do it because we were the first to make some investment in these cells. Now, let me show you just one, and this is just one from one of our published papers. So here is, now this green is the native rat motor neuron, okay? And all these little red dots are staining for the stem cells, the human stem cells that I've injected into that rat, for a stain known as synapsin, for synaptic contacts. We actually quantitated the synaptic contacts, and an average sick neuron will get anywhere between 30 and 50 synaptic contacts from these stem cells. So it's very interesting. An important point, I'm going to say it now, and I'll see if anyone asks me at the end of the talk, because I'm always asked this question. These cells cannot make axons that leave the spinal cord and attach to muscle. That's not how they provide their neural protection. They go in there, they form these contacts, they also completely, as I said, abrogate all the inflammation, and they produce a lot of growth factors. So, done with small animal models after a few bumps in the road, right? No tumors, the cells incorporate, we get great survival. So, I'm kind of like excited and thinking that my large animal model can be man because this is ALS, but it can't. And the FDA put some sense into my brain in that, I want to inject stem cells into a spinal cord of man without showing that it's safe at all in a large animal model. So the answer is no. So that's when I met, as I mentioned to you earlier, my first mini pig. So here's a mini pig who's had a lumbar laminectomy. You can see his blue scar, or you can see the area of his, not scar, it's his open wound actually, because he's just immediately post-op. And so, and interestingly, before I tell you a little bit about the surgery, it was so interesting getting to know the pigs because they had to be, MPO prior to their surgery. And when they woke up, all they were really interested in was eating. And I thought they were going to be paraplegic, really, and maybe we couldn't safely inject stem cells, but not at all. So we've done now over 100 pigs, and we have, I think, 3 or 4 publications on this. I'm just going to, again, summarize the data rather than showing it all to you. And that is that you can safely inject into both the cervical cord and the lumbar cord stem cells into the pig. And the spinal cord of the pig is very, very, very similar to that of man. And the pigs do extremely well post-operatively. There's no lasting functional deficit, and usually there's no transient functional deficit. Now, one thing that I wanted to answer is, so we inject, in the rats, there's something about injecting in the rat and then sacrificing the rat 3 to 4 months later. But there was something about injecting into the pig and then sacrificing the pig a month later that was just difficult, I thought, to handle. Plus, it was a lot of work. These pigs are really big. They're bigger than anybody in this room. I mean, these are big pigs, even though they're called mini pigs. So what we did is we ferritin labeled the stem cells so we could see them on MRI, with the idea being then what we could do is do sequential pig MRIs, which is what we did. And that was actually quite a trip in and of itself, but it was a lot of fun. So let me show you some of these data. And these are data that, again, we've just published. So here's where the pig had his lumbar laminectomy, okay, or he or she, I don't remember. And because you can use both male and female pigs, unlike rodents where you can only use male rodents usually. And so here we've injected ferritin-labeled stem cells, and maybe you can see where the red arrows are, the dense ferritin labeling. And so what we did was sequential MRIs. And so the question to me was, I was hoping the stem cells would go in there and migrate. So we'd have to do fewer injections. Wouldn't that be great? The FDA thought if they did that, it would be like, they didn't want that. That's what I wanted. Well, they didn't. So the FDA was happy. I was less happy. But what you see here is a rendition, an artist or a cartoon rendition of multiple MRIs that we've done, status post-injection. And what you can see is the cells do stay very localized in the area that we have injected. And then eventually we did a necropsy on all the pigs, or on all the pigs that we injected the ferritin label in. And here you can see at necropsy the ferritin labeled stem cells. So it allowed us to really visualize and follow the stem cells over time. Having shown in the pig, it took 100 pigs to prove safety. We were actually able to get our first phase 1 trial approved. It took us 6 submissions to get this phase 1 trial approved because it hadn't been done before, and there were just a lot of very, very good questions on the FDA's part. Very excited about when this happened, and you'll see we did our first patient in 2010, 5 years ago, because in my mind it brings an era of cellular therapies to neurology. I went to a really good talk this morning at Swedish Hospital, not your private hospital, I now know, on Alzheimer's disease. And they were talking a lot about, on amyloid buildup and how, glial cells will take up amyloid, and that's really how a lot of the monoclonal antibodies are probably the mechanism of action, is they cause increased turnover of amyloid. When you inject stem cells into an animal model of Alzheimer's disease, do you know what they do? They go right into the glial area, these stem cells. They cause the glial to be more reactive, and you see complete disappearance of the amyloid in those animal models. So these stem cells are really interesting. So I think it's going to change a lot of how we practice in the future. So let me tell you a little bit about the trial. Before I do, here's my email at the top, and here are the publications that I've summarized, except for the very top one that I'm going to now tell you the data for. So, it took about 5 years to get there, but we were able to get to where we wanted to go, and I'm happy to send any publications or answer any questions via email also. So, what did the FDA let us do? They let us do a phase 1 safety trial, and it was called a risk escalation trial, and let me explain that. So no one, here we are, we want to finally inject these stem cells into the spinal cord. The first 6 patients that we had injected had nothing to lose. They were almost essentially paraplegic. So they had little risk. But the risk escalated because the next 6 patients were ambulatory. They could walk into the clinic. They had use of their arms and legs. So if these stem cells did harm, if they actually caused injury, here are ALS patients who are already going to die, and we're going to accelerate that process rather than improve it. We began with unilateral injections, I'll show you that, and then we ended with bilateral stem cell injections. We did the first patient January 2010, and we didn't complete it. We did 18 surgeries. It took us until May of 2013. We would do 1 surgery, and the FDA would want us to stop. We'd do a second, they'd want us to stop. Every time then we did, as we advanced in the trial, we could do one a month for 3, then we'd have to stop 1 to 2 months. So they wanted to go very slowly to make sure that we weren't inducing any adverse events. And I agreed with them. I thought that this hadn't been done before, that it was very wise to go slowly. You'll see we didn't do that in phase 2. We began with lumbar injections, and I'm also going to tell you that at the end of phase 1 we did cervical injections. And so here is our phase 1 design, our trial design. And so let's start. Now, each of these letters, A, B, C, D, indicate cohorts of 3 patients. So cohort A, and I'm just going to go over one cohort, and then the rest are really self-explanatory to you as you look at the slide. So each of these syringes represents 100,000 stem cells being injected into the cord, and that 100,000 stem cells is in 10 microliters of fluid. So for the first 3 patients, they had 5 unilateral injections for 500,000 cells. If this had really hurt these patients, again, they've only had anti-gravity. We would have been able to tell, but again, they didn't have a lot of function to lose. We then moved on. When we saw that it appeared to be safe, we moved on to the next cohort. These patients got 5 injections on each side of the lumbar cord for a total of 1 million cells. Again, these patients didn't have a great deal to lose, but there was no exacerbation of their disease. Then came the first group that had a lot to lose, right? If this was dangerous or induced harm. And again, this is all about safety. And this group, these 3 patients, did fine. They walked into their surgery, walked into the clinic the day of their surgery on Wednesday, and they all walked out of the hospital on Saturday. So we then went to bilateral injections. And again, it's the same paradigm. The patients did very well. We then did a group of patients. We did 3 patients who got, 3 patients who got cervical injections. I was really sweating at the first day we did the cervical injections only. Again, patients were very functional. They did fine. And then we took patients and we did both cervical injections as well as bilateral lumbar injections, and those patients too did fine. So it was a risk escalation because each time we're escalating what we're doing and their risk is escalating.
Excuse me, did the patients have evidence of involvement at the levels that you were injecting?
Yeah, yes they did. Yes they did. But it had to be, but there were fairly strict entry criteria of in terms of like forced vital capacity was the main entry criteria. The phase 2 entry criteria were very strict. For this phase 1, it was mainly based on FVC. Yes.
The cord ends above where you're injecting. What were you injecting above?
Here, I'll show you where we were injecting. Okay. Because that we were injecting in cord levels. Here's the area that we're injecting, right? Okay, to correspond with the cord levels, and I should make that clear. Oh, and so how did we go about actually being able to target correctly? So we used MRIs on the patients, and each patient would have a very specific MRI where we would look then at cross-section, and we would measure the depth of the patient's spinal cord and its width, and we would decide to target in this particular area based on measurements. And what we did, and when I say we, this is Dr. Nick Boulis, he developed a series of very specific cannula. We call them a floating cannula. It has an outer rigid part, it has an inner pliable part, and then at the hub, what we could do is devise different needles of different sizes so we could target and hit the right area of the spinal cord for the patients. And we're targeting to hit right in here. But it's blind, I will tell you that, because while we can target all we want, it's not ferritin labeled stem cells. The FDA would never allow this, and we're not exactly doing necropsies on these patients, so it is still blind. A question I got when I gave this talk a couple of years ago that made me think I needed to include this is, why aren't you just pithing the patient? So you're injecting stem cells and the patient's breathing. Doesn't matter if your cannula floats, you're going to hurt the patient. And the reason we can safely do it is we developed a system where we actually affix the injection device to the patient. And let me explain that to you. So here's the bony structure of the spinal cord, bringing coals to Newcastle, right? And as I told you, we do a laminectomy before we inject the spinal stem cells. However, what we also do is we affix the surgical device to the lamina of the patient, anterior and posterior to where we're going to do the laminectomy. So let me show you a picture. So we will actually screw these posts into the lamina, anterior and posterior, to where we're going to do a laminectomy right here. And this is actually a picture of the surgical device, and we call this a gondola. And what we do is we take this micromanipulator and we can move it down this gondola inch by inch by inch to do our serial injections. I'm going to show you some movies and some pictures, but I think it's important to know that this isn't just me going in with a little needle and injecting a few stem cells into the spinal cord, which is sometimes the outside patient perception. Here's a typical, the operating room on a typical day. So when I started and told you it takes a lot of individuals, team science, this is really an example of team science. Not only do we have the neurosurgeons, we're also constantly monitoring the stem cells. We have a stem cell team. We have a team that's doing SSEPs to look at cord integrity at all time. And I'm somewhere there, like standing on a stool, I'm like the concert director here for this trial. It's very exciting, but as you can see, it's not. Neither is it simple. Now let's begin to look at the actual procedure, and then I'll show you a couple of movies. Okay, so now here we go. So now these posts have been screwed into the lamina, anterior and posterior. The laminectomy has been performed. We have what we call spreaders that are attached to our surgical device to open up the wound. The gondola has not yet been placed. You'll see that on the next slide. But what you can see is where you can now begin to visualize the spinal cord, which is of course our goal. Here now you see the gondola is in place. You can see the spreaders again, and now the gondola is in place. The next thing we do is the stem cell team loads the stem cells. And so how do we get the stem cells? I'm frequently asked. Well, remember I told you that made at NIH, stored still in Rockville, Maryland, okay, and they are shipped to us the morning of the surgeries. So there have been some stories about, like in one particular big Michigan snowstorm, I'm literally driving on the airport freeway with the police to get a FedEx plane that's stuck on the runway and can't get the stem cells to me. So there have been a few, kind of really dramatic episodes, but they're shipped in the morning and they're stored and kept. And they're really extremely high quality cells. So this floating cannula is loaded, and here's what the final setup looks like. So here's the floating cannula inserted into the micromanipulator, and we're ready to begin our injections. So let's go ahead and look at some injections. So the first step, you really need to completely expose the spinal cord. And we learned that through our pig surgeries, which were extremely valuable, and that when we didn't completely expose the cord, the pia, arachnoid, and dura are so tough, to be quite frank, that it would injure our needle, and we couldn't do clean injections. So it was very interesting. So here you see us completely cleaning the cord. And there are a couple of things I want you to note here, and a couple of things that I learned as a neurologist and not a surgeon. One is that the vasculature of the cord for every single patient is different. Just the way all of us have, two different eyes, nose, and mouths in this room, we all look differently. So does your spinal cord. And so has been a challenge in terms of able to do these multiple serial injections on both sides of the cord when the vasculature is so diverse. You can also I think you can beautifully see the nerve roots, see the CSF, but I hope you can also see, especially if you look maybe in this area, the pulsing of the spinal cord. Really to show you that, you can't just go in and inject. You can't inject into a pulsing cord. You have to be pulsing with the cord, which is what our surgical device allows. So we do a complete exposure of the lumbar cord. So once exposed, we really confirm our position for injection, and that is really looking at the MRIs, which are up on the screen. Plus, we do something as simple as using a ruler along with our gondola. And here you can see, again, the nerve roots. So you can see the pulsation. The cord is here, and we're going to begin injections. Here you can see the rigid outer part of our floating cannula. You can see the hub of the cannula, and we're going to begin one. In this particular case, it was 1 of 10 injections. So we're going to do 5 injections on this side of the cord, 4 millimeters apart, and then we're going to do 5 injections on this side of the cord, 4 millimeters apart. And so here's an example of an injection. So we go down. Remember now, that needle itself is a specific size based on the patient's MRI, and we go down to the hub of that needle. We pull out the rigid outer part of our floating cannula. So there's a lot of bend in this cannula, and we are now injecting the stem cells through this floating cannula. Again, note the pulsation of the nerve roots, and this is a typical injection. So, how do these patients do? Well, this is a safety trial, and we are pleased to tell you that this is a safe method. We use multiple measures of safety. One was the ALS-FRS, which is the most common tool used for ALS research, and we did not see any deterioration in the ALS-FRS, and we use multiple other clinical measurements: respiratory capacity, quantitative strength testing, electrical impedance myography, and quality of life. And we were able to show clearly that it was a safe procedure. Was there any therapeutic signal? Well, on so few patients, I feel that you cannot say that with any definite conclusion. I can just show you the data, and together we can say maybe yes, maybe no. That's why it's research. But these are the 3 patients I've elected to show you just because these are the 3 patients that got bilateral transplants. Here is their ALS-FRS prior to surgery. We were monitoring them carefully before surgery because we wanted to know the slope of their decline. And then here is their ALS-FRS post-surgery, surgery 1, and then they received the second surgery, and here's their ALS-FRS. We continue to follow these 3 patients, and now we've been following these 3 patients for nearly 4 years. We have autopsy results to share with you. So to remember those first 6 patients who were paraplegic, essentially, 5 of those 6 patients have died. So let me show you some autopsy results. So here's the spinal cord. Once we remove the dura and look at the cord, it actually looks quite healthy. I mean, from the outside, you would never know that this person had 10, 5 on each side, 10 spinal transplants, injections. And then what we do is we suction the cord. We do PCR for the cord and immunohistochemistry. Let me show you some of the IHC. So here's the cord. I think you can see it actually quite nicely. And then what we do is IHC here. Now there were no motor neurons in this cord at all. These patients were end stage. But what we did see were clusters of stem cells. It was pretty exciting to us. We haven't seen any motor neuron preservation in these autopsies, but there are really no motor neurons to preserve. We have actually done HLA PCR because we know the HLA subtype of the stem cells, and about 2 to 3% of the DNA in these spinal cords are from the stem cells. So the stem cells do survive. That's extremely important for us to have proven. But we see no other evidence except for survival and their presence. So to summarize this, because I'd like to end by showing you the phase 2, we completed a phase 1 trial. The procedure was safe and well tolerated. No evidence that it accelerated the disease course. We continue to follow 9 of these patients, and this has been since January of 2010. But still the numbers are way too small to make any claims on efficacy. And we did find DNA from the stem cells in all of our autopsy cases. So onward, we published this, and now onward to phase 2. So we got aggressive. Yes?
Do these patients have to be on any kind of medication after the...
Yes. So these patients are on, I was going to discuss that at the end, but I'll discuss it now. These patients, this is a transplant, so these patients are on mycophenolate, CellCept, and tacrolimus, FK506. Now, I'll just briefly segue and tell you that many of the patients couldn't tolerate long-term immunosuppression. Of the first 6, let's just talk about those first 6 patients, 5 of whom have passed away. So we discontinued the immunosuppression on 3 of them, and 1 had been discontinued for almost a year, and we still found robust stem cells present. So it's not like a solid organ transplant we're now beginning to understand, as are other groups. So I don't think we're going to have to immunosuppress in the next trial the entire lifetime. It's now the FDA believes, and I agree, we only need to immunosuppress for 3 months.
And Eva, I have a
Yes.
question. You showed one patient, I know this is mentioned in Annals paper, who actually got substantially better
Right.
very quickly
Right.
after treatment. What was the entry criteria in terms of the revised L-squarea clinical criteria?
Yeah, he had definite ALS.
Definite
Yeah,
ALS.
he's I can't explain him. I didn't expect it. I don't want to say I didn't wish it happened to him, because I adore this man. He's fabulous. He now sits on the ALS board, actually in Atlanta, Georgia. But he's done remarkably well. He was using it's pretty impressive. He was Kane about to use a walker before his lumbar transplants. And I've got movies of him, I'm not going to show you, where he's running in the yard with his kids. Now, I don't know. It's not what we anticipate. It's not at all what we expect. Our goal and hope is to slow progression of the disease. Yes.
So in terms of the pathology, you didn't inject the whole cord. So if you compare regions you injected versus regions you did not inject,
Yeah, we see no stem
is
cells.
there any differences?
Well, again,
For example, inflammation, TDP-43 accumulation, things like that.
we haven't done that. That has not been done. Those are a very good question. That hasn't been done. It's been done in the animals, but it hasn't been done in man. So to segue to the phase 2 design, and we're going to close with this, is we got very aggressive. One quick thing, we decided to do cervical transplantation to start with, because our reasoning was we want to really, if we're going to really preserve, we showed that we could safely do it in the previous trial. We want to preserve the large motor neurons that are involved in respiration. So we began with our first, again, I'm going to tell you, each group of patients is 3 patients, but this time they've all got an FVC of at least 70%. They are all ambulatory, they all have arm function, and only 1 patient was bulbar. And they were not supposed to be bulbar, but there was a protocol deviation there by one of the centers. So, and in this case, each little syringe now has 200,000 cells, right? In this particular case, we do 5 bilateral cervical injections, 2 million cells, and the patients do well. Onward, we then do 10 on each side. I must tell you, that day we did that, I was so nervous. I sweated out my greens completely, those surgical scrubs, and I had to take a break and go put on clean ones. But the first patient did beautifully. All patients did extremely well. We then, what we started to do is, I didn't think we could safely do more than 20 injections into the spinal cord. So what we started to do, which I knew we could safely do from the pigs and get good results, at least in the pigs, we started to increase the number of stem cells. So now each little syringe has 300,000 stem cells, then 400,000 stem cells. And then we decided in our last cohort of patients, again, to do bilateral transplants. So we did 8 million in the lower lumbar cord and 8 million in the cervical cord. Now, unlike the previous study, which had primarily been in the lumbar area, these were all cervical surgeries. So this time we had to affix our post to the base of the skull. And again, I show you that because this isn't a minor surgery or a minor procedure. Okay, but the same idea, the same surgical apparatus, laminectomy, doing injections. So let's look at some of these surgeries, because the cervical cord was clearly, especially doing 20 total injections, a clear different beast than the lumbar cord. So here now you see is clearing the cervical cord. A couple of things. It's usually much more highly vascular than the lumbar cord. Notice here the nerve roots. We have to be much more careful because the nerve roots, of course, leave at a much different angle. So you have to be very careful during injections. There's also a lot more pulsation of the cervical cord than the lumbar cord. Now, just as I told you with the lumbar cord, we then, clearly visualize the complete cord. And look how vascular this cord is, really compared to the lumbar cord. And most of the cervical cords look like that. Again, we have to be very careful because of how the nerve roots exit. And there's also a great deal more pulsations in the cervical cord when compared to the lumbar cord. However, in this particular case, for example, we're going to do 10 injections on each side of the cervical cord at 4 millimeters apart. And here's an example of a cervical cord injection. Again, notice that the angle has to be just slightly different, and the patient's at a slightly different surgical torque. But you see, again, here's our the rigid part of our cannula, and now we're pulling it out, and here's the hub, and here you can see the bend of our floating cannula, and notice how the cervical cord is constantly moving. So it was much more challenging than the lumbar injections.
How long do these take?
Oh, a surgery. Oh, that's actually a good question. We inject, each injection takes over 2 minutes. We inject over 2 minutes, then we have to leave it in. We leave it in for another 1 minute, the cannula, in case there's any backflow, and then we pull out and we move on. But it takes a lot of time to get in exactly the right position for each injection. So a surgery can take anywhere from 4 to 6 hours. So we completed phase 2 in 1 year, okay, unlike phase 3, right? Because we didn't have to wait, we could do a surgery a week. The FDA now knew that we knew what we were doing and it was safe. So we were able to do all of the surgeries in 1 year. I did not receive, I asked for approval to show you the data today because I actually now have the data for the phase 2, and I'm going to be presenting that data at the American Neurological Association in September, and it will then become publicly available. I will tell you that those data are allowing us to move forward with our next trial, which will be a very large multi-center trial. But boy, there are still challenges, right? I mean, the spinal cord is still really precious real estate. And here we are doing all of these injections. I don't think we can minimize that at all. I can tell you that in the pig, we can safely do injections 2 millimeters apart, but in man, we go 4 millimeters apart. And the question that's been asked, and it's a good one, is should we try to do our injections more closely together? We have stopped at 16 million cells in the pig. We can do double that safely. Should we consider trying to do more? In the next trial, I think I'd like to stop at 16 million cells. And then a very good question asked about immunosuppression and how long we need to immunosuppress. So don't really know. We're going to try for 3 months in the next trial. And so here's the group. I just want to point out a couple people. Here are our 2 neurosurgeons, Nick Boulis from Emory and Parag Patel, our neurosurgeon at University of Michigan. And here's John Glass from Emory. This is a multi-center trial, and Steve Goutman from U of M. A third center, as I showed you initially, also was Massachusetts General Hospital. The data will be presented at the ANA, the phase 2 data, and you can email me on Monday. If you don't go to the ANA meeting and want to know the results, I'll send you the slides, and I am disappointed I can't share them with you. But what I can share is that we will be doing the next trial. I want to invite all of you to come see me at U of M. We really enjoy having neurologists visit, and I must say it's really interesting. I threw this slide in at the end, the next slide, because I always say this, but usually no one ever takes me up on it. I have 3 neurologists visiting me this coming week because of our new football coach, and we do have the largest football stadium in the country. So thank you so much. I'm really happy to take any questions. So, yes.
I just had a curious, post-op, what is the course like? How long?
Yeah, that's a great question. You know, now I'm supposed to do something here, and I don't have my glasses on. Let's see. Was I supposed to go to notes or comments?
The meeting room.
Notes?
Down below that, right?
Below notes.
Below notes.
Yeah.
Oh, neurology, thank you. Okay, great. Of course, now I don't have my glasses, so I couldn't read the questions anyway. So let me tell you, we admit them on a Wednesday, and we put them in the ICU. They lay flat on Wednesday night, and we put them in the neurointensive care unit until Thursday. Thursday, they come out to the floor, and they go home. The latest anyone stayed until Sunday, and that was 2 patients. They almost all go home on Saturday. So surgery on Wednesday, home on Saturday. It's been remarkably smooth. And I think what's interesting, because we're neurologists, right? I'm thinking, and the neurosurgeons all along the way have said, this isn't going to be a problem. We do all sorts of stuff to the spinal cord, and it's not going to be a problem. The neurosurgeons have been right. Patients have done extremely well. Yes.
Well, that was such a nice overview of all this data.
Thanks.
In the future, do you foresee being able to use a control group? And how is that going to be an issue getting through IRBs?
That's a great question. So the question is a control group. I will tell you that I think that's important, but it really raises ethical questions. So in the Parkinson's trial, there was an old Parkinson's trial where they did a control group, and they showed no difference between the control and the stem cell graft. I think that's important. And I also think, I always tell that story of Lou Gehrig when I have time, because I think that's important. The problem is, since this is clearly a lethal disorder, how ethical is a control group? Here is one idea that may or may not take traction, is that a patient will undergo a sham surgery, which would involve just taking off the spinous processes, okay, but doing nothing else, so that they would not know whether or not they received stem cells. And after 9 months, they would be eligible to have the actual transplant. And we would follow them for 9 months, as we follow, of course, closely those patients that get 2 transplants. So that's been suggested as one idea. Whether or not that's ethical, and whether or not that's something we as a neurologic community should do or shouldn't, it's very hard, because neither should we bring to our patients a therapy that doesn't work. Just because it sounds great, it's got to work. So
in those patients, would you give them the immunosuppression, or would
yes.
you give them the placebo?
No, we would give them the immunosuppression. We have done, I didn't mention it to you again, because I wanted to show more of the surgeries. We've done a trial of immunosuppression only, which I think we needed to do, right? And you know why we did it? Because of that amazing result of that one patient who did so well. I went, holy moly, this is all immunosuppression. Oh my gosh. So we really quickly did a large trial of immunosuppression only, and there's no effect of immunosuppression.
What about as a control, doing some patients where you just do one side?
Yeah, I actually don't think, and you actually alluded to that in your earlier question, because the animal data would show that if you inject stem cells on one side, you still get anti-inflammatory effects, really robust on the other side of the spinal cord, and growth factor effects. You don't get the synaptic contacts, so I don't think that would work. There's a stem cell trial planned like that, though, out of Cedars-Sinai at LA, that I think, because they have more effects than just their very local effect. Yes?
So actually, did you do trials with the animals to just do the control for the laminectomy and see what kind of effect that had on inflammation?
Yes, we did that. And it means I probably skipped over things too quickly. I just kind of touched on it a little bit. But yes, we do multiple rodent trials, and we published those, where what we see is preservation of the motor neurons, really significant anti-inflammatory effects, a lot of growth factor production.
And with the autopsy spinal cords, did you try to find out what type of phenotype the stem cells of the remaining human cells had, whether they were still stem cells, whether they were differentiated towards something, whether they were putting out matrix?
We're doing that right now. Our neuropathologists, there's a delay between receiving the autopsies between the autopsy and the death of the patient. And our neuropathologist is not very optimistic that we're going to be able to get more information from the current autopsies. I think those are questions that we need to answer in our next protocol. At least we need to get it into the protocol so that the tissue is processed so that we can answer those questions, which we didn't.
One last question. Has anybody tried just using conditioned medium, like an acellular
Yes, yes, we did that. We did that. We published that, and there was no effect. But very good question. And I think we're out of time.
Part review question?
Oh,
Or committee?
I can't see. Oh, yeah.
On the board.
Oh, I can see the board. Thank you. Sorry. Yeah, neuroprogenitors, candidate genes, factors, and yeah. Okay. So the answer to that, let me answer the first thing. We actually, for every ALS patient that comes to our clinic, we have a biorepository that's funded where we do skin biopsies and we make iPS cell lines from any patient who's interested. And so we've been making a lot of iPS cell lines for a long time. I think that's an interesting approach, but in our hands, as in the hands of others, you can't really grow up iPS cells easily to the millions and the millions. They really have some sort of epigenetic mark, even when you de-differentiate them, that don't allow them to be as totipotent as one might want. And also remember, because they are iPS cells, do you remember at the very beginning of the talk what I showed you, what happened when you injected an embryonic stem cell type into a patient? Or into a rat, it became tumors. So there would be a lot of work yet to do to use iPS. And in terms of using engineered cells to overexpress, we actually have a neuroprogenitor line that a cortical neuroprogenitor line, so from the cortex it overexpresses IGF-1. What a surprise, right? And we're using that in Alzheimer's disease. We have not taken that approach yet for the spinal cord, but that is a very good idea. I think the iPS cell line idea is probably not so good. We've actually invested a lot of resources in that. And I guess I'll take one last question.
There's one more.
Oh, and there's one more. That's a great question. We see that in the rat, and we've published that, much to my surprise. So when the diseased upper motor neuron becomes healthier, the cortical spinal tract actually appears to remain more intact in the rat than in the presence of unabrogated disease. So maybe it's the connection that allows that. I mean, that's what we're assuming. We have also in the rats begun to do cortical injections in the ALS rats with the idea that that would be something we would eventually take to man. And those are early days. We've only done a couple of experiments. It looks like it works, but I never say yes or no after just 2 experiments. Okay, I want to thank everyone again so much. I really appreciate being here.